Welcome to The Explainer. Today, we're diving into a high-stakes, microscopic world that is hiding right in plain sight. I was absolutely floored when I learned about the intense and frankly unbelievable precision required to manufacture sterile products. We're specifically breaking down the specific requirements for the manufacture of sterile products, namely parenteral preparations, so your small and large volume injectables, as well as sterile ophthalmic preparations. The sheer engineering and discipline it takes to make these drugs safe is nothing short of incredible.
I mean, really think about that for a second. How do you take raw chemical ingredients, process them, put them into a glass vial, seal it up, and guarantee that there is exactly zero microscopic life inside? The stakes here are literally life or death. We're talking about parenteral and ophthalmic preparations that completely bypass the body's natural defenses. You're putting a liquid directly into your bloodstream or right onto your eye. There's no room for error. Absolute sterility isn't just a goal, it's the only acceptable standard.
To see how pharmaceutical engineers actually pull this off, here's our roadmap for today. First, the stakes of sterility. Then, we'll look at the environment, the human factor, processing philosophies, eradication methods, and finally, the specific workflow.
Okay, let's jump right into part 1, the stakes of sterility and bypassing the body's defenses.
So here's the biological reality. When you swallow a pill, your digestive system has some incredibly robust ways of destroying pathogens. But sterile products, like an IV fluid or prescription eye drops, they bypass those biological barriers completely. The regulations are crystal clear on this. Because these drugs are introduced directly to highly vulnerable tissues, even the tiniest microscopic contamination, whether that's a single bacterial cell, a tiny particle of dust, or even endotoxins left behind by dead bacteria, can be fatal.
So how do we build a fortress to keep these microscopic threats out? That brings us to Section 2, the environment, and designing the layers of defense.
The guidelines mandate that a facility must be divided into strictly classified clean areas, grades A, B, C, and D. You definitely don't just walk off the street into the room where the medicine is poured. Grade A is the hypersterile, critical zone where the highest risk operations happen. Things like aseptic connections and the actual filling of the vials. But Grade A doesn't just exist out in the open. It's surrounded and protected by a Grade B background environment. And then further out, you have Grades C and D acting as buffer zones for less critical stages, like washing the components before they even get sterilized.
But here's the thing. Simply having a clean room isn't enough. You have to actively defend it. You can think of a sterile facility kind of like a submarine airlock, but operating in reverse. By using what's called positive pressure cascades, the ventilation systems constantly push highly filtered air outward. The absolute highest pressure is always in the cleanest room. So if a door opens, the air physically rushes out, constantly blowing microscopic dust and contaminants away from that sterile core, completely preventing them from sneaking in.
Which brings us to a rather uncomfortable truth in pharmaceutical engineering. Section 3, the human factor, or the biggest threat to sterility.
I want you to think for a second about how much skin and hair you naturally shed every single minute without even noticing. It is a lot. From a microscopic perspective, humans are basically walking biohazards, and in a Grade A clean room, a single shed skin flake carrying bacteria is a catastrophic event. That is exactly why the regulations dictate that only the absolute minimum number of personnel are ever allowed in these clean areas. It's why there are insanely strict rules requiring specialized, sterilized body and hair coverings. And it's why anyone with an open lesion or even an abnormal skin shedding condition is strictly barred from handling these materials. We are the contamination risk.
So how do we solve the human problem? Well, the regulations highly encourage advanced engineering solutions that just remove us from the equation entirely. Take blow-fill-seal technology, for instance. This is incredible. It's a single, continuous, automated machine that takes raw plastic granules, melts them down to blow the shape of a container, fills it with a sterile liquid, and seals it shut, all in a fraction of a second, entirely inside a sterile barrier. By using closed systems and isolator cabinets like this, you physically separate the human from the critical Grade A zone.
Okay, we've kept the humans out, but how do we actually make the drug itself sterile? Let's look at Section 4, Processing Philosophies, the 2 paths to purity.
The first, and honestly highly preferred method, is terminal sterilization. This is where you put the drug into its final container, completely seal it shut, and then basically cook it using intense heat in an autoclave. It's virtually foolproof. But there's a catch.
Many biological drugs like vaccines or certain hormones will be completely destroyed by that heat. So that's where aseptic processing comes in. This is the incredibly difficult task of sterilizing the drug, sterilizing the glass vial, sterilizing the rubber stopper, and then assembling all of those pieces together in that hyper-clean grade A environment without introducing a single microbe during the process.
Whether we're sterilizing the final vial or just the components, the rules are strict. This is Section 5, Eradication Methods, our weapons against pathogens.
So, looking at our arsenal, moist heat, or steam, is the absolute gold standard for terminal sterilization. Now, for dry powders or oily liquids, you'd use dry heat. If a product is superheat sensitive, ionizing radiation or gases like ethylene oxide might be brought in. And for aseptic processing of liquids, we push the drug through microscopic filters that literally trap the bacteria in their tracks. Oh, and here's a really fascinating detail. The guidelines explicitly ban ultraviolet irradiation as a terminal sterilization method. It just isn't reliable enough to penetrate the final product.
But listen, here is the absolute most important rule in this entire framework. It is never enough to just turn on the autoclave and cross your fingers. Every single sterilization process has to be scientifically validated. Manufacturers have to use biological indicators and temperature probes to prove beyond a shadow of a doubt that those lethal sterilizing conditions reach the absolute coolest, hardest to reach center of the batch for the exact required amount of time. In this industry, if you can't document it, it literally didn't happen.
So what does this actually look like in the real world? Let's check out Section 6, the specific workflow, taking a look at the anatomy of a parenteral facility.
If we trace the life of a vial through one of these facilities, you'll see a very strictly ordered, logical flow. It all kicks off in water management, turning regular water into highly purified water for injection. At the same time, over in the container prep area, glass vials are being meticulously washed and dried. Next, the active drug solution is prepared and filtered. Then, all these elements converge in the grade A filling and sealing area. Once sealed, the product might move to terminal sterilization. And finally, every single batch goes into quarantine and undergoes a rigorous visual inspection to check for particulate matter or defects. The whole physical layout of the building is deliberately designed to keep this forward momentum, completely preventing any mix-ups between unsterilized and sterilized materials.
And if you want a perfect example of just how intense this final stage is, look at this number. For containers that are closed by fusion, think of those glass ampoules where the tips are literally melted shut. The regulations demand exactly 100% integrity testing. Not a random sample, not 99%. Every single ampoule must be tested for microscopic leaks to prove the sterile seal is completely perfect. It is a true, undeniable zero-tolerance policy for manufacturing errors.
Which leaves us with a pretty wild final thought. We've seen that the regulations require an absolute fortress of pressure cascades, validation protocols, and strict zoning just to protect the product from, well, us. As technologies like blow, fill seal, and advanced robotics become more and more sophisticated, it really begs the question, how long until humans are completely removed from the sterile manufacturing equation entirely? Thank you so much for joining me to explore the incredible precision of pharmaceutical engineering in this explainer. Until next time, stay curious.